My name is Yasmin Berumen Martinez, and my project is on the novel salivary protein apolipid-foreign in plant interactions with pea aphids. My mentors are Dr. Karen Gomez and Lonnie Iron, and I'll explain this, so don't worry about the title. So what are pea aphids? Pea aphids are agricultural pests. In the literature, they're referred to sap-sucking insects, and this is due to their unique feeding mechanism where they insert their stylet down into the phloem of the plant. And their stylet is just like a straw-like structure, and it's very flexible. And it reaches down, and the phloem is where the nutrients are in the plant. So they want all the nutrients up so they can survive and deprive the plant of all these nutrients, killing the plant ultimately. Additionally, there are vectors to many plant pathogens and viruses. So when they're feeding, they transmit these pathogens and kill vast agricultural patches. They also reproduce asexually and sexually. So depending on the season, they can switch their reproduction. Additionally, when they're stressed, they switch their reproduction. So they go from sexually to asexually when they're stressed, and they also develop wings. So if they're in the crop and it's dying, they develop those wings and they can move to another crop, infecting more. So apolipid-foreign is the other big part of my project. And apolipid-foreign, we called it LIP in our lab, and it's suggested to do with lipid transport and innate immunity, and also essential for feeding and colonization. And colonization just means that they invade that crop, kind of stay there, make that their home. And I say suggested because we don't know anything about apolipid-foreign. It's novel. There's not a lot of research on it. The way that we found out is through their neighbor, apolipid-foreign 3, which does have to do with lipid binding and immune defense. So what we're trying to do with LIP is knock it down. And to explain knockdown, I have to give you a little biology crash course on centrodogma, which is this picture. So we have DNA, which then makes RNA, which then makes our protein. What this means is that we're working in the molecular level in RNA. We will insert double-stranded RNA of LIP in RNA, which brings up like red flags to the cell, and they start attacking any code of LIP they see. So it's not shown in the aphid because the protein stops right there. The protein isn't made. So I already talked about a bit about how pea aphids are agricultural pests because of their unique feeding mechanism, their vectors, and their reproduction system. But the problem with this is that we have pesticides, but they target beneficial organisms. So what we're trying to do with apolipid-foreign is that it's specific to pea aphids. So targeting it in pea aphids, it will only target in pea aphids and not target other beneficial organisms like our pollinators, which are needed to grow fruits and pollinate other crops. Additionally, pesticides and insecticides are shown in our food, so in our produce. So we don't want that either. And so we're trying to find an eco-friendly and alternative for pesticides and insecticides. So going along with that, my research question is finding the role of apolipid-foreign to then find an alternative or eco-friendly way to target pea aphids without it getting into our food. Additionally, my second research question is on the knockdown effect of apolipid-foreign in insects and the reproductive rate and survival success. So with this, we hypothesize that knocking down apolipid-foreign will inhibit feeding and colonization of our plant and also the overall survival of the pea aphids. So the first step is making that double-stranded RNA so we can knock down the gene. So we use about 10 aphids and then put that in liquid nitrogen and then extract RNA from them to synthesize for cDNA and then synthesize double-stranded RNA. Then we apply this to a new aphid that's alive and we apply it in the dorsal area of the aphid, so on top of it, so that it can go through. We also add nano carriers so that it can go through the cell for better effect. And then we transfer these to our experimental plants. So the first thing we're doing is looking at the expression of LIP to ensure that I did do the double-stranded RNA and it was successful. So with this, we also look at our control, GFP, which is a green fluorescent protein. And all it does is just illuminate the P aphid so it glows. If you ever seen those glowing fish, that's what it does. So it doesn't target anything. For this, we have 3 time points, 0, 24 hours, and 72 hours. And these time points were decided from preliminary data in our lab, which show that LIP is more active during this time point. We have 3 plants per time point and then 5 aphids per plant. Our second thing is looking more into the role of apolipophilin. So we're looking into mortality and reproductive success, what overall it does, and how it affects the P aphids. We have 3 repetitions for this, 3 plants per repetition, and then 5 aphids again. And we look at this for a longer period of time. So we look at around 5 days. And we count for the nymphs and adults. And nymphs are just baby aphids that they produce, and how many adults have survived from the 5 initial. So in our first experiment with expression, we collect the heads of the aphid during these time points, and then we extract RNA and synthesize for cDNA to then do qPCR and analysis. For the feeding, we also obtain the colony mass of the aphid to see how well they're feeding. So usually the ones that are feeding better weigh more, and the ones that are feeding less weigh less, and do an analysis of this. So preliminary data, and this is for mortality and reproduction success. We can see that GFP and LIP over time, the aphid survival of LIP is lower than GFP. You can see it as it's going throughout our experiment. And then for this one, you can see the specific time points that it starts to go down. So the first one is 0 hours. So they're both in the same level because we just did it. And then this is 24 hours. So GFP, our control, is right there, and then LIP goes all the way down. And also at the end, we have GFP up over here, and then LIP all the way down there. So we almost had 100% mortality success with that one. So for future directions, looking at the gene expression and reproductive rate and fitness of the overall aphids, we're doing a NOVA test and a student t-test, and also doing a Tukey's post hoc, which just groups all the time points together to see which ones are significant. Our expected results are a full knockdown of LIP and higher mortality for LIP than GFP. This is my timeline. So I already did sterilization and transplanting. So the next big things is doing my double-stranded RNA drop on the aphids. And I will be doing that November 29th, and then counting where I have all my data collection done would be December 4th. Acknowledgements, I would like to acknowledge the Gomez Lab, Dr. Karen Gomez, Lonnie Ivrin, the collaborators at CSU and UNC, and then the undergraduate research grant for funding this, and an NHS grant, and also the McNair program for everything they do. These are my citations, and then thank you. Any questions? Uh, they live like for a couple weeks. Yeah, so they're not like dead. Like the GFP ones that we found still lived. They shouldn't have died, but we think it's possibly due to the CO2 we used when doing it to put them to sleep. So yeah. So we did like look at it throughout other organisms to see if it like affected them. That's why we chose lip because we saw that it didn't. So the double strand part of it, it's hard to put it in the dorsal level of the aphids because they're squirming around. So we did it based off of that and also how expensive it is to make double stranded RNA. So any more questions? So yeah, we're looking into, so first we're trying to find the role of it to see if it could be used as a alternative pesticide and insecticides and hopefully group it to that research and help that like bring it up a bit. I want to say.